This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 333, recorded on April 9th, 2015. Hi everybody, I'm Vincent Raccaniello, and you are listening to TWiV, the podcast all about viruses. I'm back in Nashville, Tennessee. I went home and I came back after a week. And I'm visiting Vanderbilt University again. And this time I have 4 students with me who are members of the MSTP program, the Medical Scientist Training Program at Vanderbilt University. And we're going to talk about what it's like to be in this program and what you do and all that, which we've never really talked about on TWiV. So it's cool to have you. Let's introduce all of you all the way down the table there. He is a 4th year graduate student, Bobak Parang. Welcome.
Thank you. Good to be here.
And right next to Bobak is a 3rd year medical student, Megan Freeman.
Hello.
Welcome to TWiV.
Thank you very much.
And next to Megan is a 2nd year graduate student, Meredith Rogers. Welcome to TWiV.
Thanks for having me.
And finally, but not least, a 1st year graduate student, Ben Fensterheim. Welcome to TWiV.
That's it. Thank you.
And you guys are all MSTP members. And what I want to do is talk about what it's like to be in the program and so forth. But I want to start by asking, what is medical scientist training program? I always thought of them as MD, PhD programs. So you get both degrees, right?
Yes.
What is MSTP? Who wants to answer? Let's start with you, and then we can go down the line.
Okay. So the MD, PhD program is something that many schools have, where they award both degrees to students that want both degrees. The NIH, independently of these individual schools, deciding that they were going to award both degrees, wanted to promote the awarding of both degrees to students. So they created a medical scientist training program, or MSTP, that awards money to institutions like Vanderbilt to train students as MD, PhDs.
Okay, so not every MD, PhD program is going to be an MSTP.
Right.
Exactly.
Right. How many MST programs are there? You know?
I think it's something like 40.
Yeah.
40?
Yeah.
And how many MD, PhD granting institutions? More?
I don't know, like
Yeah, a lot
100
more
or
than that.
something.
Yeah.
There's a lot more. Just it's
Really anyone that had a medical school and also PhD granting.
I mean, the
institution.
MSTP, you basically do them at the same time, right?
right. So
Whereas-
it's integrated. And,
Yeah, but other people can go get a PhD and then later decide they want an M. So there are people but that's not a formal program, right?
No,
Yeah.
but there are
So-
schools who have formal MD, PhD programs.
And they're not MSTP. 'Cause
Right.
They don't
just
have funding.
You know, they're perhaps newer, or for whatever reason they haven't gotten NIH funding for that program.
So when did NIH start this MSTP?
I believe it was in the 60s.
Okay.
And I think Terry Dermody, our director, will be disappointed if these are all incorrect facts. But, I think it was in the 60s out of sort of the recognition that there were a shortage of physician scientists, and so the NIH made it a priority to fund them. Now, I think Vanderbilt really went through some cycles. I think in the 80s there were some funding issues, but I think in the 90s is when the program really picked back up in terms of membership, like size of each class. But I think the growth of these programs having 6, 7, even up to 15 students per class is fairly recent in the past 10 years or so, 15 years.
So the program arose because we needed more physician scientists, essentially. So people who do research, but from a clinical standpoint, who could also take care of patients and then go pipette, right?
Yes.
Exactly.
Yeah.
Like people who
Yeah.
could appreciate, translating their science to bench to bedside, really.
So every MSTP program, or I guess every MD, PhD program has a director, right?
Yeah.
You need to have someone, and here it's Terry Dermody. Right?
Yes.
I don't know who it is at my institution. I believe we have an MSTP program at Columbia. And I know Wash U has one. They're a very good one.
Yeah.
There are many others as well. How many are in this program? How many students?
I know we're over 100, so
Wow.
they're 8 years pretty much in the program, right? So you'll do 2 years of medical school and then 4-ish years of PhD, as long as it takes. And then the traditional model has been to do 2 years after that
Okay.
of medical school also. So if you think about, you have 8 years of students that are in this program. I know we're over 100.
Yeah.
New classes, we're shooting to get 15 students per year.
So it's an 8-year program, no more.
Can be more.
Our new curriculum is actually, so the new students that are coming in are enrolling in curriculum 2.0, which is a medical school creation, and we've adapted the MSTP to that. So actually, if you were to start this year in the MSTP, your time to graduation would be 7 years because of the new curriculum. But
If you take a 4-year PhD,
Which is the
right,
average.
so the average will, but the average will move from 8 to 7. But then this depends on, of course, your PhD.
Right.
Yeah.
Right.
So you do
Yeah.
2 years of class initially, then you go into a lab for 4 years,
Yeah.
or maybe less, right?
Right.
Could happen. Miracles could happen.
Yeah.
Yeah.
And then you go back, there's no more class, but you do hospital things, right? You do what's called wards or clinics.
Yeah.
I don't know what it's-
Clerkships. Right.
Yeah, clerkships, right. So you go into different services and you spend 2 years. Maybe the last year you do something freelance or something? Is that, because I know a lot of medical schools, the last year you can do research or whatever you
So the
want.
idea is that you can really tailor your 4th year to your interests. So as part of the new curriculum, they're offering more courses now that can be tailored to your interests. So
So Ben, you're a first-year grad student, so you finished 2 years of your classroom. Bobak, you're a 4th-year grad student, so you're about to go back.
Yep.
Into
Defending in
the war.
June, actually.
Very
Yeah,
good.
Very excited. Yeah.
And Megan, you're a 3rd-year medical student, so you finished your PhD.
I did. I finished in July.
You defended?
Yes.
Congratulations.
Thank you.
Very good. We'll talk about your work.
Okay.
And Meredith, you're a 2nd-year grad student.
Yeah.
So you've got some time. All right. Let's find out about you guys, where you're from and so forth. Where are you from originally, Ben?
I was born in Washington, D.C., and I've pretty much lived around there my whole life.
Went to high school?
I
there?
moved out to Rockville, Maryland. So I went to high school in Rockville.
Big move, right?
Well, I was little, so my parents had this really awesome brownstone in Foggy Bottom, DC, which is such a great area.
Yeah.
And then my mom got pregnant with me. I was the first child, and the steps in the brownstone were very, very steep. They were very, very steep, and they were worried that I was just going to fall and smash my head. And so they said, we're going to sell this brownstone and move out to the suburbs, which is what they did. They sold the house for a few $100,000 at the time. 20 years later,
It's worth millions.
we're living right in the suburb. The house is worth millions.
Well, it's
So
not
he
yours.
moved so he wouldn't hit his head.
Oh, well.
So I'm from around that area.
Where'd you go to college?
So I left and went to college at the University of Michigan in Ann Arbor
Yeah.
for undergrad. Yeah.
And were you a science major, I guess?
I studied both neuroscience and philosophy. I started out as philosophy and then ended up getting more and more involved in neuroscience. So.
I don't know. Do you know, what's his name? Here we go. My memory is fading. Mike Bishop and Harold Varmus. Do you know Harold Varmus?
Yeah.
So he used to be director of NIH. He's
Yeah.
currently NCI director, but he was a literature major in college.
Huh.
I went to Columbia, actually. And then from Michigan, you came here,
Yes.
applied to this program,
Yes.
got in. Okay, Meredith, where are you from? I know where you're from. You told me, but you
I
can
did
tell
tell
us
you
again.
before. Yeah, so I'm from the Los Angeles area. My parents still live in the house I grew up in, so. was
In
there
the
for a long
suburb of
time.
LA?
Yeah, the suburbs. Like very few people are actually from Los Angeles, the city. And so went to high school there, did my undergrad at Tufts University in Boston, and then came here straight out of there.
This is the Tufts out in Chestnut Hill, right?
It's Somerville,
Somerville,
Medford.
yeah, the outside of the city. Because
Yeah.
the medical school is in the
Right,
city.
yes, there's two campuses
And
there.
then came here from Tufts. And Megan, what about you?
I am from Lexington, Kentucky, where I also went to high school and college at the University of Kentucky. So I lived there a long time, and then came here straight after.
Were you a bio major in
I
college?
was a biotechnology major, which was actually in the ag school. So that was kind of an interesting.
Meredith, what did you major in?
I majored in biology.
Okay.
Yeah.
Tufts. I don't know anyone at the Tufts outside of Boston, but I know people inside Boston. They have a great virology faculty there. And Bobak, where are you from?
I'm from Knoxville, Tennessee, so just a couple hours east of here. Went to high school there and then did my undergrad at University of Pennsylvania, and then came straight out of my senior year to here.
Yeah.
So kind of coming back home of sorts, but Nashville's a very different city than Knoxville is also.
Yeah. Were you a bio major at Penn?
Yeah, I was a bio major, and I was really enjoyed evolutionary biology, so that was sort of my sub major, and I did some work in some evolutionary biology labs there. Well, one lab for the whole time I was there, and that's really where I kind of fell in love with research and that whole community of it.
Penn, I was just at Penn 2 weeks ago.
Yeah, you mentioned
for
that talk. So.
symposium. They have a great virology faculty.
Do they?
In fact, Mark Dennison, who's on the faculty here, worked with Susan Weiss, who is a coronavirologist there at Penn. But there are lots of other great virologists, really good. One of the few schools with tons of virologists in the country. So Ben, what got you interested in, not Ben, Bobak, I'm sorry. I'm down the other way.
Yeah.
What got you interested in science, medicine, or both? At what point did you decide on science and then a dual program even?
Yeah, I think, so my interest in science was pretty much really formed, especially in biology, at my freshman year of high school. And we had this great biology teacher, and when we got to the subject of evolution, we were forbidden to be taught about humans and evolving from apes. And being a 14-year-old and being told you can't learn something, I really wanted to learn about it. So I went home, and my dad, who's a mechanical engineer, really liked reading this author, Stephen Jay Gould, who was a paleontologist at Harvard and wrote a lot of popular essays, and I think Natural History is the journal. But he gave me a couple of his books, and there's just wonderful essays on evolution and science and a lot about the history of science. And so that was kind of the igniting point. It was just sort of fell in love with these concepts and intoxication. I think evolution is also probably the most poetic, beautiful concept in science. And then sophomore, junior year, I took a genetics class, and then the idea of DNA was just so mysterious, and it was that whole idea kind of, at that point I knew I wanted to do science. And then when I went to Penn, it was sort of assumed, I think for a lot of people in my time, that if you were interested in science, you were pre-med. The idea of research wasn't a big thought. But I started getting involved in this evolutionary biology lab there and did basic stuff that most early undergrads do. I was washing dishes and helping make LB auger plates. And I was talking to a senior graduate student at the time, and I was telling her, she was asking what I wanted to do, and I said, I'm not sure, I really love this research, but I also really like medicine. And she was like, well, have you heard there are these MD PhD programs? And I said, I don't know what you're talking about. And sort of just from talking to her, I just started to research them. And I spent almost my entire sophomore year writing to MSTP directors, and even, especially at Penn, talking to some MSTP students and asking what that was like, what that entailed. And then after talking to so many people, I decided my junior year that I was going to apply to those. And I think someone has mentioned this. I think it was Liz Dong in our program mentioned this, saying that a lot of the reasons that why MSTPs are in MSTP programs is because we're just very indecisive people, and we like both things and we can't pick. And I think that's very true for me, at least. I just really enjoyed the research part and really enjoyed the medicine part as well.
So did you apply also to single degree programs?
No, I actually only applied to MSTP programs, which was, in retrospect, it's a really tiring process because I think traditionally MD programs, you interview for about a day. I mean, you fly in the morning, you're out, you have a couple interviews. But the MSTP interviews are like 3-day affairs where you come in on a Thursday, you eat dinner at Terry Dermody's house. Actually, Megan was our student host, was my student host. So you have student hosts, all day Friday you're interviewing, and then Saturday you're still here and you do a tour of the city. And this is pretty much how it works in most of the programs. So they're pretty exhausting, and especially as a 22-year-old, and you think you know a lot about your science, but you're also meeting these people very famous and very intimidating. So it's a grueling interview process.
How many did you apply to? How many programs?
I applied to probably 20, and then as I got interviews, I started to sort of cancel some later on, and then I found out I got into Vanderbilt in January, and I still had several more interviews, but I wanted to come here as soon as I interviewed, and I canceled everything else and pretty much accepted off the bat. So I think I ultimately went on 6 or 7 interviews, but I applied to probably 20 places.
Cool. Megan, how about you? How did you get interested in science?
Yeah, so I went to a math, science, technology high school, Magnet, in Lexington, and as part of kind of our senior project, we were expected to work in a lab at UK and kind of get exposed to that. So I remember very little about that lab experience, aside that I learned how to make gels and I learned that science was a thing that happened that people could do in labs, and that I liked being there and I liked being able to ask people questions and kind of how that whole interchange works. So then I go to UK and was a biotech major, and that summer I thought maybe I would get a job as like a barista or something. But then I got cast in a community theater production and I needed a day job so that I could go to rehearsal at night. And I knew that I was going to need another lab project kind of towards the end of my tenure. So I decided I would go lab hunting, and was really lucky and landed in Becky Dutch's lab for 3 years, who studies paramyxovirus fusion, which was an incredible experience for me. So there's a MD PhD program at Kentucky, a small one, and a student in that program was kind of my direct supervisor in the lab, and he spent the better part of 3 years saying, "So Megan, you know, it'd be a great idea is if you do this MD PhD thing." And Bobak says that we're indecisive, and I think that we're also kind of naturally curious as a population of people.
That's a better way to put it.
Right, and I just think that we're people that like learning how things work. And how humans work. And I think that anyone that will sign up to be in school for an extra 8 years has to have that really as a quality to kind of keep you going. So I just like to know how things work, and science was a really good way for me to be able to do that. And I also really like talking to people, and I thought that science was a good way for me to be able to do that as well.
I had Becky on a TWiV a couple years ago. I don't know if you saw it.
That was a live TWiV.
Her hair was making havoc with the mic.
That's how I knew, because
She
I was
said-
at ASV. I was there.
You were there. So
Yeah.
she said, "Should I just hold it?" I said, "No, that really wouldn't work for you to have your hand." So we had a lot of hair noise, but that happens. It's funny that you would have a similar problem.
I did.
If you worked with her. So do you think that MD, PhD students are different from MD students, people who are just interested in an MD degree?
Yes and no. I think that for a lot of us, we want to take care of patients, but there's a lot of memorizing that has to happen in medical school. One of the things that people tell you about it is that you have to learn another language. So you have to learn the word for trouble swallowing, and you have to learn
Is that dysphagia?
It is dysphagia.
Do you know what sternutation is?
Sternutation. Do you guys know that one? I don't know that one.
What is that?
It's sneezing.
That's
Actually, we have a
excellent.
listener who's a pediatrician in Cambridge, MA, and she told us that.
Okay, that's a good
I
one.
love the words.
So you'll have some professors that will tell you this is a $10 word and then give you the
Yeah.
word, and you're supposed to keep track. But I think aside from learning just all of those things, we as a cohort tend to be more interested in how things work.
Yeah.
Instead of just like, this is the dogma of what we know about something so far, we're the ones that will ask questions that people won't necessarily know the answers to.
Yeah, I interview, not only do I teach at a medical school, but I interview students for the MD, PhD, but not for the MD program because they're not interested in basic scientists, right? And it's very interesting. I also teach undergraduates, and I see this curiosity as undergraduates, and then by the time they go to medical school, it kind of is dissipated. They're not curious about basic science anymore, which is too bad.
Yeah, I completely agree. I think there are very different groups of people, and I think they become even more different as an individual goes through medical school, and then as an individual goes through the MD PhD program. And I think a key part of that is the graduate school portion. I think it really does change you, and it changes your perspective on a lot of things. Because at medical school, you're in there, and you know what you need to do, and you do it, and that's how you get your readout is your grades, or you get a pass on the clerkships. But I think there is something, there's just something deeply humbling and unique to the PhD portion and getting acquainted with research and the failure and having such ownership of it. But I think there are definitely more curious people, and not to ruffle any feathers, but I also think, at least in my experience, I think MD PhDs tend to be more well-rounded people than MDs. I think MDs are very goal-oriented. This is just in my limited experience, probably small sample size, but I think the kind of person who's interested in a problem and wanting to identify solutions to it a lot of times also someone who has other interests in life, and it's not, everything isn't just in pretty one-dimensional.
Yeah. I have to say, the students I interview, I'm just awed by, because they're like perfect MCATs, 4.0 or 4.3. They've done so much volunteer work, it makes me tired to read their resumes. It's just unbelievable. I mean, I had a guy who had perfect MCAT scores, right? And just amazing. And he had graduated college at 14 or something like that.
Jeez.
So I've had a few in my lab, and they're really, I enjoy having them because they have this nice combination, and they're very smart too, and they can work well. Meredith, what got you interested in science?
So I don't come from a science family. Like my dad's a lawyer, my mom's was a librarian. She's retired now. But I think so. My older brother, when he got to high school, he was taking some like science classes and was kind of talking to me about it and was excited about what he was doing. And so that really kind of influenced me when I got to high school to really pay attention to my science classes and to really, be curious about that, because my older brother, who I looked up to so much, was also interested in it. And then I guess when I applied for college, I was kind of toying between English, like creative writing, and science. And so Tufts for me kind of offered options for both. And then I explored both, took both English classes and biology classes, and ended up my summer after my freshman year in a lab back in LA at City of Hope. And after talking to kind of different PhDs and postdocs in that lab, and somebody said, well, if you're the way they kind of posed it was a more practical sense of, if you're interested in doing this, you should do MD PhD because it's a lot easier to get money.
Yeah.
But ultimately, I'd come into it thinking I wanted to do a PhD, and I guess we were working on HIV in that lab, so it kind of, got me aware that there's this field of science where you are doing things that are directly impacting patients and stuff, and so that was kind of really cool to me, and I just kept exploring both and found a way to do both.
Did you apply only to dual programs? Mm-hmm. And
Yeah.
you as well, Megan?
Yes, only dual programs.
So what would have happened if you didn't get into any?
Yeah.
You didn't think about that.
Should ask them.
As you
I mean,
did.
you apply widely, I guess. I mean, I think I went to 15 interviews.
So are some schools easier to
So,
get in than others
yeah.
then? I would guess there's a range.
I mean, it's like
They're all
any
competitive, right?
school is, there's a range of how competitive one school is versus another, and you have your reach schools and your. I mean, I don't think any MSTP program is easy to get into, but
Yeah, there's no guarantee. Like in college, there's no easy. What do they call it? A sure
yeah.
school.
Yeah.
I stratified my schools based off of reach and I guess safety. And the first thing I heard back from any school was a rejection before the secondary from my most safety school.
Oh no.
That was the first thing I heard back. And I was like, well, gotta get a new job. And but then luckily I ended up at a great MSTP.
Yeah.
And so it's hard to really know, but you do try and stratify. And
Yeah.
So how did you get interested in science?
I kind of took a circuitous route to get interested. I was not very interested in school at all in high school, but then had a really great English teacher that inspired me to study philosophy. So I picked up my grades enough in high school to go to Michigan, which was awesome because then I could study philosophy at Michigan, which is one of the top philosophy departments in the country. And I studied ethics, and I became very interested in why people choose to do the things that they do, and what the right thing to do is. And it was this big problem that I was thinking about a lot, and I figured I should get it into science to help me solve these problems. And so I joined a lab with Josh Burke, who is a behavioral electrophysiologist, and he studies the basal ganglia, which is what we think is part of the brain that allows you to make quick action decisions. So whether or not to move your left hand or to move your right hand, or whether to make an impulsive decision this way or that way, is all sort of made up by the basal ganglia, which is really interesting because that's not the cortex of your brain where you're thinking. So
This is like cloud computing, right?
Yeah, it's very confusing because it turns out that the cortex is actually more just rationalizing what the basal ganglia is doing for you. So instead of you having this top-down, you tell your arms to move, it's more like your basal ganglia saying, like, we kind of want to do this, and then your cortex goes, oh yes, let's do that. So it was very interesting for me from a philosophy standpoint to study this, and the lab was so cool. So much technology. We would implant recording electrodes that could record from like 128 neurons simultaneously from a rat brain, and it just blew me away. So I worked really hard for 3 years in that lab, knew I wanted to do science after that, and then when my project was ending, I wanted to see it through, not just publish the paper, but then do something with it afterwards. And my postdoc mentor was an MD PhD, and he was a neurologist. And he said, yeah, we implant these things in people, and we can do this in the clinic if you wanted to take this project and pursue it, that it could actually help people. And I thought that was so awesome that I decided, based on his advice, I took a year off, and during that year I applied to MD PhD programs.
And only
MSTPs, yeah.
Wow, well, for you people. Maybe that's why you were picked for TWiV. Is there anyone that didn't just apply to it? Probably there are.
I'm sure there is, but after you start a program, you aren't really talking about what could have been or whatever.
Sure, of course.
I was on the admissions committee for a little while, so I can tell you that a vast majority of our applicants apply only to MD PhD programs.
It's that focus, right?
And I think one of the things too I've heard is they want to see that you are very committed to the MD PhD program, and not that you're going to maybe start the program and then bail out of the program, or you're going to start it and realize it's not for you. And so one way to get at that is to see where are people applying. If they're kind of applying all over the place and they're not really sure, then maybe that's a sign that they're not.
Sure.
Yeah, you wonder if they're trying to get 2 free years of medical school or something.
Yeah.
So as part of this program, medical school is paid for? Is that
Everything is, yeah.
everything? Which is different from regular medical school,
Yeah.
right?
It's an enormous benefit, yeah. I mean, we're very fortunate for that because we get a stipend, a graduate school stipend, essentially our entire 7 to 8 or 9 years we're here, and we don't have the debt problems that are pretty crippling for a lot of medical students.
Yeah. But
So-
of course, that's not why any of you applied to the dual program.
No.
Well, no, it ends up not being worth it because you have to spend 4 years doing research, and in those 4 years, you could make significant gains on your debt if you just were an attending physician. Because essentially they're making up ground as an attending physician. And then for the rest of our lives, if we really do want to pursue science, we're going to take a little bit of a pay cut from just a full MD physician. So it ends up not being worth it if that's just all you wanted.
So let's talk a little bit about your science. Bobak, you're a 4th year. You're about to defend, right?
Yes.
What have you been
That's
working
something I tell
on?
myself every morning. So I'm in the cancer biology department. I'm in Chris Williams' lab. And we sort of half our lab does studies inflammatory bowel disease, and the other half studies colon cancer. And pretty much my primary dissertation project is a gene that was discovered here, which is blood vessel epicardial substance, but the acronym is BVIS. And it was discovered here by David Bader's group as being important in cardiac development. And before I had joined the lab, we had found that BVIS was underexpressed in cancers, colon cancers. And if you put BVIS back into colon cancer cells, it induces this epithelialization, and they become less proliferative, they become much more normal. And so my project, really, when I joined the lab, was to identify a mechanism, some sort of cellular mechanism. And we have this yeast-2 hybrid list identifying a number of proteins that BVIS interacts with. And so I kind of picked one of those and tried to identify it, and we're trying to get this manuscript published. It's been sort of all over the place the past few months. But essentially, I've found that, well, we really as a group have found that BVIS is interacting with the oncogene MYC, which is overexpressed in 50% of cancers and really thought to drive proliferation. And we found that BVIS can interact with MYC, protein-protein interaction, and it promotes MYC's degradation. And so when you lose BVIS, you lose one of the regulatory mechanisms of MYC, and it can kind of drive a lot of tumor
So is
genesis.
BVIS mutated in colon cancer?
The promoter is hypermethylated, and so there's a big CPG island in the promoter, and so it's downregulated because it can't get expressed. There are mutations in some of the cancers, but for the most part, we see that it's lost at the transcriptional level. Just cells can't make it. So yeah, really essentially we have the mice that don't have BVIS, the knockout mice, and we've put them through cancer protocols, and they have higher tumor number, and then we've looked at a lot of patient data sets to see if BVIS is expressed or not, and it's pretty much uniformly in a lot of epithelial cancers, it's downregulated. So that's kind of been my primary work is really trying to.
So are you thinking that BVIS could be a therapeutic at some point?
Yeah. So the difficulty is, turning on genes that are methylated requires methylase inhibitors, and these have global changes and they're very toxic. Right. So it's tough to look at that from a transcriptional standpoint. We do think that. There are a subset of patients who have inflammatory bowel disease because they go through these recurrent cycles of inflammation. So these are patients with ulcerative colitis or Crohn's, and they get diagnosed very early, sometimes in their teens, and they go through 30, 40 years of these inflammatory bouts. And because of that, they're at a much higher risk for developing colon cancer than the normal population. And what we found is that in a subset of these patients, if you do a biopsy of any section of their colon, and you look at the BVIS promoter, its methylation status, the BVIS promoter is hypermethylated in patients who do have cancer. So we're trying to sort of view it as being able to identify the methylation status of the BVIS promoter as a biomarker for patients, and it would tell us that they have cancer somewhere in their colon, and then they need to be examined by colonoscopy pretty carefully and thoroughly. The big thing with colon cancer with all these patients is if you catch it too late. If you catch it early, it's an enormous cure rate, but if you catch it late, stage 1, identifying stage 1 colon cancer is a 90% cure rate because you can remove it. Stage 4 is a 10% cure rate. So if we can develop any tools that allow us to identify a risk and really thoroughly perform a colonoscopy and monitor these patients, it is a big difference in their survival outcome. So we view it we think the utility of BVIS right now is a biomarker. In terms of a therapeutic target down the line, that'll be tough.
So how would you do the assay? What are you thinking about? It would have to be a methylation assay?
Yep, exactly. So this could be in patients who have ulcerative colitis or inflammatory bowel disease. They go through more frequent colonoscopies than the average population. So what we're envisioning is that a 35-year-old person who has ulcerative colitis gets a routine colonoscopy, and during that colonoscopy, you take any piece of their colon, and then you can run methylation analysis for BVIS.
I see.
Because the alternative is that the gastroenterologist with the scope surveys the whole colon and examines for any cancerous lesions. But the colon has folds, it has ridges, and it's not the easiest thing it's not something that is, it's not a science to it. It really does require a good exam, and that's tough. And a lot of times, these patients, for any of your older listeners, and my dad tells me this, but the prep for the colonoscopy, you have to drink about a gallon of salt water the night before to, quote unquote, flush the colon. And that is a horrible experience. And if patients don't do it, understandably so, there's a lot of stool in the colon that blocks a good exam.
Yeah. I know I've been through quite a few. Because both my parents died of colon cancer.
Oh, I'm sorry.
I have to go weekly for a colonoscopy. No, not weekly. Very frequently.
Yeah.
I mean, I think I should go every year, but my gastroenterologist says that's too much. But I'm worried that it'll miss it, or.
Yeah.
He told me they don't grow that quickly, so 5 years is good enough.
Right. I think that's
true.
I think that's true. Yeah. These are pretty slow-growing things.
So my mother had a colonoscopy in November, supposedly clean, and she died in May from metastatic colon
my
cancer.
goodness. That's
So I must have missed it, I guess. I don't know. But it's very. Why does inflammation predispose to cancer?
Yeah.
All the proliferation that's going on?
It's a great question. That is an area that is studied by so many gastroenterologists and really all cancer biologists. But yeah, it's thought that there is a lot of both the process, the inflammatory infiltrates themselves will promote proliferation, and then the epithelial processes of recovering from a wound or any damage. That restitution recovery process itself promotes proliferation and a lot of migration. And then there's also growing kind of area in the field is that all these inflammatory changes also induce widespread genomic methylation. So you have this chronically, you get a lot of methylation changes that are permanent. And there's a huge new area now that is kind of taking over the GI world, and that's the microbiota and how these are influencing these processes. And this is the tip of the iceberg, but it's becoming increasingly clear that just depending on your bacterial species in your colon can protect from certain colon cancers or promote inflammation. And maybe a lot of this is our microbiota is just disrupted. And a really interesting study to point out, there's actually a paper that just came out in Nature a month ago or so, but they put mice on a number of very common emulsifiers that are found in our own foods, preservatives, and put them at pretty small doses that were thought to be representative of what we have. And they not only changed these mice's microbiota, but they developed metabolic syndrome, they developed inflammatory changes in their gut. I mean, very widespread issues, and they linked a lot of it to the microbiota, these emulsifiers having this disrupting effect. So in terms of why inflammation drives these, it seems to be many factors.
There's probably also a role for the virome, the gut virome.
Absolutely.
A paper was just published suggesting that people with Crohn's or IBD have altered viromes. Whether that's associated or causative, we don't know, but that's another area. The microbiome is easy to study because you just sequence ribosomal RNA, right? But the virome, you have to do genome sequencing. So.
Right.
But I think that we shouldn't ignore that.
Yeah.
It'll be very interesting. So Megan, what do you do?
So I earned my PhD with Mark Dennison,
Right.
who's a recent guest on your show. We study coronaviruses in the Dennison lab. So examples are SARS and Middle Eastern respiratory syndrome coronavirus that has been ongoing. So we're really interested in ideas of viral evolution and also viral cell biology. So when I joined the lab, we had this live cell video of a cell that was infected with murine hepatitis virus, which is the BSL-2 model that we use in our lab to study coronas, that late during infection started doing these membrane ruffling events, which were really dramatic. And this was right before I joined the lab, and Mark would show this video to everyone and say, hey, isn't this cool? What do you think it is? Hey, isn't this cool? And he, talked to a bunch of folks and finally started talking to some cell biologists, and they said, that looks a lot like something called macropinocytosis, which is a type of cellular drinking where actin in the cell is dynamically reorganized so that this ruffle can extend from the surface of the cell and sweep in whatever is in the extracellular fluid and internalize it. So when I joined the lab, my job was to see if this was macropinocytosis, and if yes, then why were coronaviruses inducing it? So I spent a lot of time doing that, and it looks like, yes, it was macropinocytosis. And the interesting thing about that in the context of this virus is that many viruses recently have been shown to use that as an entry mechanism into the cell. So you can imagine that it would be a really sneaky way for a virus to get into a cell. A lot of cells are doing this anyway. It's how we survey our immune systems a lot of the time.
A receptor is still needed, though, right?
There's not specifically a receptor that we know about that triggers macropinocytosis. It's mostly through EGFR and like the RAC and PAC kinase systems. So a lot of viruses can activate that, and then as the cell starts scooping in things non-specifically, it's a quick way for them to get into the cell. But we were seeing this at somewhere between 4 and 8 hours post-infection with coronas. So what ended up happening is I discovered that it seems to be more likely that coronas are using it as a mechanism to spread from cell to cell. They form syncytia in cell culture and also in some animal models, which is a giant multinucleated cell. So it basically encourages the cell to fuse to its neighbor, maybe for increased resources as the virus is replicating. But we think that macropinocytosis is another way that if this this viral protein can hitch a ride on this actin arm, that it can infect neighbor cells without actually being released and being vulnerable by attack from the immune system.
So it's produced in the cell and then hangs out on the arm and gets push to a new cell? Is that the idea?
Yeah, that's our idea. So a lot of work towards the end of my PhD showed that it was related to the fusion protein of the virus, which often is how we think viruses get into cells, right? So we know that towards the end of infection, the layer of the cell on the outside, the membrane, is studded with spike protein, which is the fusion protein that's just alone and independent of other things. So we think that it actually might be interacting with EGFR at the surface to stimulate this ruffling and to be able to infect neighbor cells and fuse with them.
You're probably familiar with vaccinia being propelled
I am.
on similar tails of actin into neighboring cells, right?
Yeah, really cool work.
There's some really beautiful movies of that
Yeah.
from Jeff Smith.
Vaccinia is one of the first ones that I read about that used macropinocytosis for entry to some of Ari Holanis's work.
So in your case, it's not so much for entry, but
More
getting
likely
into the
for
neighboring cell.
exit or for neighbors and not for entry.
So all your work was in cell culture, right?
Yes.
Is there any way to see if this happens in an animal?
Yeah, I thought about that a lot. We don't actually, interesting story with MHV. So MHV is the scourge of many mouse facilities
That's right,
in the scientific community.
Oh, yikes.
And as everyone that works with mice, like.
Killed our facility.
Yeah.
Absolutely killed it.
So the Vanderbilt facility actually has a policy that we're not allowed to use MHV in the mouse facility for understandable reasons. So
You need your own facility.
yeah, it's not something that we were able to do here. We have some collaborators that could work on it. Something that I had thought about is I spent a lot of time using inhibitors of macropinocytosis, one of which is called EIPA, which is a type of a milleride that is supposed to inhibit it. Also, different EGFR inhibitors seemed to inhibit macropinocytosis. The problem from a systemic perspective is that EGFR is important for a lot of things
Yeah,
in
of
one's
course,
cells,
of course.
so it would be really non-specific. So that's not something that we worked out quite yet.
So you're now finished with your lab work here in the clinics now, right?
I am.
You miss the lab?
I miss the lab a lot. I also miss reading about what's happening in science. So I'm so thankful for your podcast
Yeah, you
because
should
I
just
can
listen up.
I can fit that in, and I still know a little bit of what's happening in the virology world. I spend a lot more time now reading about specific problems that my patients have and trying to learn from them, which has been refreshing and really, really great in a different way. I think I'd forgotten how much I liked clinical medicine because I was so focused on how much I liked basic science. So my first rotation back in medical school, I was just like, oh man, I really like doing this. This is really cool. So it has its pros and cons.
Yeah, that's why they picked you.
Yes, because I like these things, I suppose.
Indecisiveness.
Curiosity.
Curiosity.
Meredith, what are
That'd
you
be
up
our
to?
title.
Yeah, so I work in John Williams' lab, not the famous composer. But so we study
Does
human.
he like Star Wars at all? Probably not.
We're kind of a nerdy lab, so I think so. Or geeky, I don't know what the right word is. So we study human metapneumovirus, which is a paramyxovirus. It's the second leading cause of lower respiratory illness in children, behind RSV, which is its kind of more famous cousin. MPV was only discovered in 2001, so it's kind of a slightly smaller field versus RSV, which has been around for a lot longer. And so what I'm doing, my project is kind of at the interface of immunology and virology. I'm working off of the work from a previous student who was also an MSTP student, John Erickson, who he discovered that, so to back up, so MPV and other respiratory viruses, they cause this phenomenon of T-cell impairment in the lung. So you have a T-cell response to the virus, you have T-cells that recognize that virus, but then they don't actually make their cytokines, they don't do things that a T-cell normally does to fight an infection. And so
Is that because the virus is doing something?
we think it's really kind of an immunoregulation sort of thing. I mean, it's been shown by enough viruses that it's maybe something that viruses exploit, but it's been shown by so many viruses that it's difficult to think there is a virus product that is, causing that to happen. And so, so T cells become impaired, and so our previous work in the lab showed that the inhibitory receptor program, cell death 1 or PD1, as it's well known now, partially mediates that impairment. And so when you block PD1 or you have mice that are genetically ablated of PD1, that they clear virus faster, that they have a better T cell response. But it turns out that that isn't the whole story, that only kind of mediates part of that impairment. So I'm kind of coming in to try to understand this a little bit better. I'm working on regulatory T cells, as well as kind of doing a little bit of work on PD1, understanding the ligand for that, and the fact that this ligand is expressed by so many different types of cells, and trying to figure out, is there a subset of cells where that ligand is important? Is it possible that that process of T cell impairment is occurring before that T cell encounters a virus-infected cell?
So there are other viruses that infect the lung and they induce CTL responses, and this doesn't happen, right? So why, or does it in many other cases?
So, I mean, it's been shown for flu and for RSV as well.
Yeah.
We think that, this doesn't often happen for acute infection. People think of T cell exhaustion as a chronic infection problem, so a barrier to being able to cure HIV and hepatitis C and those sorts of viruses. And so we think that maybe just the lung is a special place. So you kind of, it's important to limit your immune response in order to be able to keep breathing when you're infected. And so that this is maybe a mechanism for kind of homeostasis within the lung during an infection. But whether the viruses themselves, have exploited that's, another question.
I think the CNS is also a special place in that sense, because it doesn't want to have a big immune response, right?
Right.
So it has other barriers to prevent. And most viruses don't get in, but when they do, then
It's bad, yeah.
then you have big trouble, right? And so when flu gets down into the lung, it can cause serious problems, possibly in part because of this. But there is, I always thought there is some CTL-mediated damage in the lung. That's part of the reason why you feel lousy, right?
There's so different cytokines are part of your, inflammation response. It's difficult to say, because most humans recover from their virus and then you have no reason to look in their lungs. So it's hard to say. It's clear that some of the damage is being done by the virus, and that some damage, for things like flu especially, is being done by the immune system. So kind of where that meets is a really interesting question.
So do you do any of this work in mice, or is it all? Yes.
Pretty much everything I do is in mice.
So if you take out T cells from an infected mouse, many of them don't have functions that lysis or something, right?
Yeah. We've mapped epitopes for MPV, and for one of our immunodominant epitopes, you'll have say 10% of your CTLs recognizing that epitope, but then maybe only a few percent of them actually produce interferon gamma or things like that.
And does this recover eventually, or does it persist for the life of the cell?
So I guess that's a hard question to ask, because of, we're taking snapshots of cells, so you can't then put that cell back in the, I guess it'd be kind of a cool thing to do, to study that, but, it seems like just kind of longitudinally we've done studies out for a number of months that if you then re-challenge, that those mice or those T cells still, have some level of impairment. Whether that's the same T cell as before, it's not quite clear.
So it would be interesting to look in people, right? You can do lavages and get cells out.
Yeah, that's one of the things that I'm trying to do, and I actually we're trying to get samples. Well, our lab is actually moving, but what we had been trying to do before kind of that whole process started is to try to get some samples, because patients that are in the ICU, they get tracheal washes, and so that's an easy collection of cells. But the problem with that is you need to then store those cells for long enough to be able to not have dead cells to look at.
That's my view of the physician scientist. You go do a lavage, and you go back in the lab and you do flow cytometry.
Study the fluid.
Yeah.
You guys can do that.
Yeah. I would really like to do that. It's just finding good collaborators to help us with that will be important.
So the whole lab is moving?
Yeah, so John is becoming the division chief of Peds ID at University of Pittsburgh Medical Center. So we'll be moving at the end of June.
What do you think about that?
I mean, it's a little bit exciting. It's a little bit terrifying.
So you're going to be part of that MST program?
No, I'm actually, so that's the interesting thing. So I'm going to stay a Vanderbilt student. So basically I'll be doing my research there, and I'll actually come back here
Okay,
for committee
right.
meetings and stuff like that, and then return to
the
finish
last 2
medical
years?
school. Yeah.
It's not so bad. But you just have, the living issue is you got to go there and then you have to come
Yeah.
back again.
I mean, it's a cool city, I've heard, so I'm excited.
There's a great coronavirologist at Pittsburgh, Carolyn Coyne.
Yeah.
Go look
Yeah.
her up.
John's good friends with her.
Good, really good. Yeah, she was on a TWiV also. I guess everyone
It's
is
just
on TWiV,
keeps
right,
coming.
eventually. So Ben, you're a first year, but you must be already pipetting, right?
Yes, definitely pipetting. Actually, we started a little over a year ago today, so I've just finished up my first year there, and I'm in an immunology lab after all that talk about neuroscience. I actually switched as soon as I started medical school, because it turns out I didn't know anything about immunology, learned it, and just fell in love with it. So, and it was a little more clinically relevant for me at the time. My clinical interests are more in critical care than they are in neurology now. So I'm in an immunology lab, and my boss is an anesthesiologist. His name's Ed Sherwood, and he works with just all the patients that he works with in anesthesia and in critical care anesthesia are patients that are very high risk of infection. And so these include patients that are just going in for surgery, burn patients, which is a big population he's working with, and patients in the ICU for other medical reasons. And his job is to keep them alive while they're at the stay at Vanderbilt, while they're staying here at Vanderbilt. So our lab, as an immunology, he's trained in immunology, and so our lab is trying to figure out ways to reduce the risk of infection in these people. And that's becoming a more pressing problem with antibiotic-resistant organisms that are growing. So we have taken the approach to try and boost the immune system with an immunomodulatory drug that lots of people are probably familiar with. The origin of this immunomodulatory drug is lipopolysaccharide, which is found on the bacterial cell wall of a gram-negative bacteria. It is very, very immunogenic. It promotes a humongous immune response, so much so that it can kill you if you have it, so it's not safe for use in humans, but we use a non-toxic derivative of that, which is basically lipopolysaccharide with a phosphate group removed, found on one species of Salmonella bacteria called monophosphoryllipid A.
It's an adjuvant used in
It's,
vaccines.
yes, it's an adjuvant used in the hepatitis B vaccine and the HPV vaccine.
Right.
It's commonly in combination with alum, called
it's
ASO4.
It's a TLR4 ligand, I believe,
Yes,
right?
so just like LPS,
That's what it tickles, okay.
it binds the TLR4 receptor, and it promotes an immune response that is similar to LPS, but not so overwhelming that it's going to actually kill you. And so a while ago, people were proposing to use monophosphoryllipid A for this purpose, boost the immune system a little bit for a short period of time while these people are in the hospital so they can resist infection better. But the field actually stopped pursuing the drug in the early 90s because LPS and MPLA produced this phenomenon called endotoxin tolerance, which is a phenomenon where if you're exposed to LPS, like during an infection or something, you don't respond to further doses of LPS. And this is something that they realized and they thought was very bad, because if you don't respond to further doses of LPS and their response measurement was cytokines, then you are going to be susceptible to infections. So if you look at people in the hospital that have just been septic, they will not respond to LPS, and that's called endotoxin tolerance. And then you see these people go and get secondary infections. And you know, the reason why they're getting those secondary infections is because they cannot respond and they cannot provoke an immune response to bacteria. But nobody actually tested whether LPS alone can predispose you to an infection. They all were looking at this phenomenon of endotoxin tolerance, which was, can LPS decrease your response to further doses of LPS? But no one actually checked an infection. And my boss and a couple other groups checked this about 15 years ago and found that mice and other larger animal models like sheep that are given an injection of LPS are way better off against an infection. They seem to clear the infection in a much quicker time point and prevent it from, like a local infection, let's say, from becoming a full-blown septic infection. And so this was very confusing, and still people today don't believe that this is a thing, where if you give a mouse lipopolysaccharide or MPLA, that they will be better off against an infection.
What's
Because
the interval? How much time can?
So-
is it quick or?
So it works within about a day. So you can get protection within about a day, and we don't know exactly how long it lasts, but it's probably around 7 to 10 days after the injection. So where I come in is trying to sort out this phenomenon, which is these mice, which are previously treated with something like LPS or MPLA, do not make cytokines in response to bacteria, but they clear infections very quickly. Lots of different bacterial infections. They don't discriminate between gram-negative or gram-positive. They can even clear fungal infections very quickly. And so figuring out why or how these animals can clear the infection without making cytokines when the infection arises is sort of where I come in. So I'm studying macrophage responses. So macrophages actually have the TLR receptors on them and are responding to the monophospholipid A. And observing what they're doing. And then someone else in my lab, Julie, is looking at the neutrophil responses. So it turns out if you inject something like MPLA, it basically wakes up the cells that they need to go and fight something, but nothing is actually there. So they turn on, they become activated, they float around if they're a neutrophil in the blood, or they crawl around if they're a macrophage, but they don't actually use their energy that they're building up to kill bacteria for anything. And they'll stay in that state for a few days, and then they'll just return to normal if nothing happens, or if you're a neutrophil, it'll eventually die. But if an infection arises, they're ready, they're waiting, they're right there, they don't need to release cytokines, and they can just phagocytose the bacteria immediately. So that's really, at least what we think the mechanism is. It's just cell activation, and so I'm working on the macrophage response.
So do you know if that effect is TLR4 dependent?
So the original effect is, so if you get rid of the TLR you will not see an effect from LPS
So
or
the
monophospholipid
protection, in other words,
A.
the priming of the max by LPS, that's TLR4. If you do it in
Yes.
TLR4 in no mouse, you don't see it.
Right. The priming is TLR4 dependent. That's TLR4 is the main receptor for MPLA.
So what are you trying to figure out specifically? What the macrophages are doing particularly, or how they're activated?
Yeah,
or-
so what we're really interested in is how can you activate a macrophage and keep it in a primed state for about a week and then have it return to normal after that? And how does that process happen? And we think what's going on is the metabolism of the macrophage is actually turning on. So when macrophages become activated, they switch their metabolism from normally you run glycolysis, a citric acid cycle, and oxidative phosphorylation. But when you become activated, you switch your metabolism to be primarily glycolytic. So the macrophage can make certain effector molecules, like metabolic intermediates, so it can phagocytose, et cetera. And that metabolic program can stay running for a pretty long period of time after the TLR4 ligand, whatever it is removed. So the macrophage can stay in an activated state by keeping its metabolism running. It won't undergo apoptosis, and that allows it to act fast as soon as the bacteria arises. So I've gotten deep into macrophage metabolism as a mechanism for that. And so, of course, my boss is an anesthesiologist, and we want to move this into the clinics as fast as we can. So we're trying to get clinical trials up and running for monophospholipid A in burn patients, as well as patients undergoing elective surgery for something like
Right,
colon
sure, yeah.
cancer or something like that.
Because does it tap into your philosophical bent as well?
You know what? I haven't actually taken it there, but cells are very interesting, but I haven't really found a very. There's not a great brain inside of a cell. They seem to do what you tell them to do every single
Yeah.
time.
It's surprising that the cells stay active so long. You wouldn't guess that. So that's pretty cool.
Yeah.
One could wonder if there's free will with these cells.
Yeah, right. We will. So let's talk about some other things. Bobak, what's been the hardest part of being in this program?
I think I will say I think we have I haven't been a student in other programs, but I think we have one of the best and most supportive run programs. Our leadership staff is just tremendously supportive. I mean, they treat us so well that we really only have to focus on what we have to do in the lab or focus on our grades in medical school. I think a tough part is, you start off in med school with a class of medical students, and you go with them for 2 years, and then you leave, and you're in the middle of grad school, and they go and they match, and there's this very celebratory mood, and they go become neurosurgeons and physicians at these big places. And so it's a little sad seeing them go. It's definitely a big part of your social component. And that tends to be in a point where you haven't really published a paper yet. You're kind of a nobody in the lab. You're getting things running. So it's tough to see people go. But I think the uncertainty of grad school and of research, I think becoming acquainted with that and accepting it is a really tough thing. And just taking ownership of your own projects and essentially being okay with failing and not letting that get you down too low in the lab, and being okay with 90% of the time things don't work. I think that's pretty rough. But when you get those highs, you try to make them count and make that keep winning your sales for a long time. But I think those are the two toughest parts. I think getting to the PhD can be pretty tough, and I think you start off this program really young, and you really kind of grow up through it too. I mean, you start off in your early 20s, and most of us pretty much hit 30 when we leave, and there's a big transition that happens in your social life and your own life. But it's been really fun. I think it's been very challenging, but I've never experienced any other phase of my life where it's been as fulfilling and as enjoyable.
Megan, challenge?
So I agree absolutely with everything that Bobak just said. And I have one thing to add. So when you're at the end of your PhD and you feel like you know everything about this one tiny piece of the world, two things happen. The next day you leave, and then other people start to learn more things about that, and you have less time for it, so you feel a little distant from it. And you also have this feeling of kind of autonomy and ownership over what you were doing. People in the lab ask you for help, you teach people things, and then you go back to the bottom, and you just don't, you do what you're told, and you don't get as much ownership over projects and directing teams and that kind of thing. And I really missed that at the beginning of this year, so that's something you have to kind of get used to. I think it's better, but in the beginning that's kind of rough.
Meredith.
Yeah, my classmates just matched, so I'm in that.
Oh no.
I'm in that halfway point here.
Yeah, it gets better.
Yeah. But I think, for me, coming from non-science family, and my friends in college, they're doing economic stuff and stuff that's just really not science in the way that I'm doing. So it's kind of, I feel like sometimes like I'm speaking a different language when I'm talking to people, and especially trying to tell my parents what I'm doing and why I'm going to be in school for, why I have a bit more time, and go to Thanksgiving at my grandfather's house, and I will have to remind people, no, I'm still in school and I'm still got a few more years. But at the same time, I really can't imagine doing anything else.
How about you, Ben?
Well, something that was very tough for me was transitioning to graduate school, something I did last year. My whole life had previously been planned for me because it had all been school, and I had to be here at this time and do this homework for this day. And that was it my whole life, until day one of graduate school, where my boss, we had a conversation, and he said, "What do you want to work on?" And I said, "That's a great question. I should go read." And so I read a lot about it, and I came back with some ideas, and he said, "Oh, that sounds like a great idea. Go and do that." And so I would go back and I'd look at my Google Calendar, and it just had nothing on it. And I had to say, well, okay, how am I going to do this thing that I just thought of one day? And so originally I overplanned my week. I planned too many experiments that were probably all a waste of time anyway, but I planned them over-planned, and I was staying very late, and then I thought that was a bad idea, so then I started under-planning, and it would be 2 o'clock in the afternoon, and I would have nothing to do, and that was a problem. So probably time management was something that I really struggled with at first and had to learn about, and I think it's probably the most valuable thing that I've learned since I've been in graduate school, because that's how everyone's life really is. Once you get into the real world, it's not planned for you. You have to really decide on your own what to do. So it's probably been the biggest
So
thing.
on the other hand, what's been the most satisfying aspect of being here?
Well, so far, at least in my limited experience in graduate school, having experiments go well, especially if you have made the hypothesis that they were going to go a certain way, is just exhilarating of an experience and could keep me going. Just one experiment going well could probably keep me going the whole year. So that has been really great. And from the medical school side, I've actually really, really enjoyed learning about medicine, and I don't want to say that I enjoyed the STEP exam, because I did not, but when I was done and I reflected on how much I knew or had to know to take the Step 1 exam, I was very, and I think everyone should be that does, very proud of the amount of information that they know.
So what's the Step 1 exam?
So in order to be a licensed physician in the United States, you have to take 3 STEP exams. Step 1, Step 2, Step 3. And the first one, Step 1, you take after your second year of medical school, and it's mostly assessing your basic medical knowledge, anatomy, pathology, pathophys of disease. Step 2 is more clinical care. How would you manage this patient? And then and that's taken in your 3rd or 4th year, and then Step 3 is taken in residency. So Step 1 is I haven't taken the other 2 steps, but Step 1 is traditionally thought of as the most rigorous and intense exam, and requires the most amount of basic knowledge in order to take. And when you do it, you really feel like, I could do this.
This is a nationally standardized exam, right?
Yeah, it's the overall exam is the United States Medical Licensing Exam, the USMLE.
It's like the MCAT for medical students.
Yeah, but
not
the
too,
LSAT.
folks take like 4 or 5 weeks off to just study for this exam, and then you sit. It's what, 8 hours?
Yeah, it's an 8-hour test.
It's a long test.
Yeah, I think I saw 3 different rounds of PSAT students come into the room that I was at. It was like 10th graders go and take the PSATs, and they come in and they're there for 2 hours and they're sweating, and then they leave, and then another one comes and sits down, and then they leave, and then another one comes, and the whole time you're still taking the same test. So,
Wow. Meredith, what's the best part for you?
I mean, it's partly the people I've met, I guess. I mean, I've, made some really great friends, and, made some really great connections with the different people in my program, and in, different scientists and things like that. But also I think it's just really cool that at the end of this, somebody's going to be willing to pay me money to do something that I really love to do. And I think that we're really fortunate to be in that situation.
Megan.
Yeah. Kind of like Ben was saying, I had this one span of maybe a month or so where I designed like 3 or 4 experiments that got at this problem in different ways, and every single one of them worked.
Oh, nice.
And that had never happened to me ever before, and I was so excited about it, and it's all in my paper. It all made the cut, so that's exciting. So that was really great. And I mean, also, like Meredith said, I've met some great people. And I just think it's really cool to think about like how many people I know that are scientists. And we'll sit around, and apparently like the discussions that scientists or doctors have at dinner are different than other people, which will be apparently or very apparent when like there's a spouse that's maybe a computer engineer or something like that, and you end up grossing them out at every single meal conversation that you have, and you don't notice that you do it.
Yeah, my dad was a physician. He never talked at home at all. He was a surgeon and never really said anything at the dinner table. I guess that's why.
Yeah, that might be why.
Yeah, I think my happiest moment is just hearing Megan say it's possible to have a month of successful experience. But I think similar. I'd had this hypothesis since I started the lab that Beavis was interacting with this oncoprotein MYC, and it took me about 2 years of experiments, and I had kind of shown peripherally that this was potentially possible, but I had the first sort of definitive evidence. It was actually last Memorial Day, and I saw the blot, and it was just the most thrilling thing. And that has been wind in my sails since for about a year. But it was just finally feeling like you had learned something that was significant. This is not field shattering by any means, but the previous experiments and projects, were successful, but they're pretty incremental. And this was something from day one I had sort of this hypothesis I'd formulated, and just seeing that band was incredibly thrilling. It was a very exciting moment. So that was kind of the pinnacle of graduate school.
Cool. Yeah, science is great in that way. So let's look forward. What's next for you?
So I'm defending again. Let me repeat that so it's people know it's on record. And then that's in late June, and then I'll probably wrap up some projects and take at least a week off. And then medical school's the next 2 years. And then for me, I will apply. I'm pretty interested in medicine. I'm pretty sure I want to do medicine. So I'm going to apply to programs that are called short tracking programs. So these are for physician scientists who want an extra year of research instead of in residency. So I'll do, hopefully, 2 years of a medical residency as opposed to 3, and then I start a fellowship a year early. And that, instead of doing that 3rd year of my medical residency, it gets tacked on to the end of my fellowship for research. But I think, 15, 20 years from now, I would really like to be at an academic hospital. At this point, I'm pretty sure I want to run my own lab, but I just want to be in the academic community, doing something with research in medicine. It's just like they're saying too, the kind of the community of people is just really fun. And you're always meeting very talented people, very interesting, and it's always fresh. People are always coming and going, which has its cons because you're saying goodbye, but one of the pros is you're always saying hello to new people. And it's just very exciting. There's always an energy. So I want to do something in this setting 15, 20 years from now.
It's one of the reasons I like having a lab that everyone leaves eventually.
Yeah.
So if there's problems, it doesn't matter.
It goes away.
Just gotta wait it out.
What about you, Megan? Where are you going?
Yeah, so I am getting ready to finish up year 3, and then year 4 is the last year. So I'm actually married to another MD PhD, and we'll be entering the match in the fall, and it'll be a couples match. So we have to kind of coordinate all of that. So the idea is that you apply to X number of programs. I'm interested in pediatrics, and then would like to do a short track like Bobak was describing, but into infectious disease. And then my husband wants to do pathology and cancer. So we need to find a place that will be good training for us, and then we'll have some people that we might like to work with and that sort of thing. So that's kind of coming up. And then I get to take more STEP exams this year,
Right.
so that's coming up too.
So in 10, 15 years, what do you see yourself doing?
Yeah, I'd also like to, I think, have a lab that looks at questions of relationships between viruses and cells. And particularly, I'm interested in kind of zoonotic pathogens that emerge and cause lots of problems. And then would like to treat children in the hospital with infections.
Well, when you have your lab and you're working, let us know. We'll have you back on TWiV.
Sounds good.
Meredith, about you. What's next?
Well, I guess short term, move to Pittsburgh and then come
Yeah.
back.
Yeah.
And then, I am interested in continuing in this infectious disease kind of area, possibly with adult medicine. But you never know. I hear that third-year medical students, their eyes get open to a lot of different specialties and a lot of different things that we may not have considered previously. But research-wise, I'm definitely still interested in infectious disease and immunology, which luckily pretty much everything that can go wrong with the human body has some element of immunology mixed in. So there's relevance to a lot of different stuff. And then, probably would want to do a short track type residency, and also, trying to find an academic medical center. I think that's really, the most streamlined place for us to be able to do both is at a medical center that is also doing research. That's really kind of what it's for.
Are there any requirements of you as an MSTP student? Do you have to do something after you finish? Nothing. You could just go sell pizza if you wanted, right?
We could go into private practice, or there's nothing binding. which is why I think there's so much energy that goes into the admissions process to identify someone who really wants to do this.
So what are the numbers? Do most people do research careers like you've been talking
The
about?
majority
two-thirds.
do academic medicine.
Yeah.
Interesting.
So the the group that goes into private practice, which I think is like maybe 15-20%, is always sort of viewed upon negatively in the academic community.
We can't
because-
talk about that.
Yeah, we don't talk about them.
Yeah,
But
but
you know,
they have a research view of things that other physicians don't, and I think that's good, right, in a way.
Yeah.
So if they're going to practice, at least they can interpret things differently. And, you know, when a patient says, why do I need to get this vaccine, maybe they're going to be better at explaining it than other physicians.
right. Also, in their defense, I think when you're 22 and you think you want to do something for 8 years, sometimes you just don't know. Also, you know, and your interests change. So I don't think these are people who are selling out consciously and saying we you know, sticking it to the man. I think there's some genuine
Yeah,
there's some
of course.
genuine changes in mind.
I had one of my best students was a MD PhD student, and she was fabulous. She went to UPenn and did a research residency, and the chief of ID said she was the best person he'd ever seen. She quit. Just went home to raise her kids.
Oh, none even practice. No kidding.
She's brilliant.
Oh no.
She had twins, and she could not go back to work. And then by then it was too late, and she didn't want to go back. I hope she's not listening. But well, she knows who she is, but she's just so wonderful in the lab, and apparently clinically she was really good too. You know, so it happens. It's life. It gets in the way sometimes. What about you? Where are you going? So you're early on, but
Well,
in the
yeah,
future.
a little early. I just selected my committee, so I will be qualifying in October this year for the Path Micro and Immuno department here at Vanderbilt. Looking way to the future, what I would write on right now, what I would hope to do and write on an application would be something in critical care. Definitely taking care of patients that are in the ICU or around ICUs, like in the OR. I really love anesthesia, and that's one of the reasons why I started working with my PI. I think I still like to do that. I like clinic, it's very interesting to me, but when I'm in the OR shadowing, which is all I can do right now, I feel like I'm more in my element there. I like it there a little bit more. It's the environment, the types of things they're doing. I also love physiology. That was my favorite part of medical school for sure, was learning physiology. So I hope to do something along the lines of critical care, physiology, maybe anesthesia, maybe critical care medicine, I'm not sure.
It would be interesting to get you all back in like 10 years and see where you
Where
ended
have
up.
we gone?
But you'd be too busy, and I'd never be able to get you in one room. See, this is beautiful because you're kind of relaxed and contemplative, but you know, in 10 years you're going to be crazy. I don't have an hour to give to you. I'm sorry.
No comment.
So the last thing, I have one more question, but I want to ask you. So we have two women here. Is this representative of the program? 50%?
We might actually have a few more women than
Yeah.
we do men.
Yeah, we had a few years where there were fewer women, and like my class has 4 women in a total of 13. But I think it's evened out again now.
I think a
So-
couple years ago we had a graduating class that was all women too.
Yeah.
I think Terry always
Yeah.
referred to them as class of.
It's like the mighty women of the M4 class
Yeah.
when it was them. Yeah.
So is the medical school class similarly balanced? They make an
Yes.
effort to maintain balance, right?
Yeah.
Yeah, I think so.
And I don't know if you know about graduate school. Is it the same or?
I think it's the same.
I see so many women in
Yeah,
our department. I don't know.
At least in the cancer department, I think there's probably more women graduate students than there are men.
Yeah.
But I don't know IGP. I don't know our whole department in general, the whole graduate school.
So the last question I have is, think about people who are out there listening and they may be interested in dual programs. Give them some advice, Bobak.
I would say if you have an interest in research or medicine and you're confused about it and you have any glimmer of interest in an MD PhD program, I think the best thing you should do is talk to people. So if you're at an institution or you're around people who are current MD PhD students, those are great people to talk to. I would talk to as many as you can, and then also something to not be afraid of is to email directors of programs and ask for a meeting. I did that, and people are so much more willing to meet than you think they are. They're very kind, despite the fact these are very busy people. But I think it's critical to talk to as many MD PhD students as you can, and don't be afraid to email a course director or program directors. These are all people who have the best idea of what it is because they're living it, and they can give you the best understanding for it.
Yeah.
Maybe.
I think talking to people is very important, and also just identifying good mentors for yourself. I've learned so much from there's a few people that really stick out in my mind as people who not only science mentors, but I knew kind of had my best interest going forward, and I was really able to have good discussions. Becky is absolutely one about kind of what I should do and what would be a good career path for me and that kind of thing. And something that I know a lot of folks get discouraged about is science funding and being able to make it in science these days. And it's so encouraging to have people that you respect tell you things like, if I can do it, you can do it. I know you'll have enough ideas. So those kinds of things are really encouraging and wonderful. And also emailing current students. I've had peripherally, someone would work with someone that I knew and was interested in MD PhD, and I'll get random emails from people that I've never met asking about programs and kind of how is it to be an MD PhD student. I need help applying. Like, what do I need to add to my application? What's a good strategy? And folks are always happy to help out.
Great. Meredith.
Yeah, I mean, I think it's valuable to kind of try to work in labs where there are people, whether it's your PI or just other people that are somehow involved in some sort of clinical aspects of medicine, to understand what is this career like for people. We say, I want to do medicine and I want to do research and I want to do both at the same time, but to see if somebody's actually living that life is really important, to understand what it's like and, to talk to them about it. I think getting down to it and talking to current students once students are thinking about applying, I think is important, to just understand where to go and what to prioritize when people are looking into this program.
So can people contact you if they listen to TWiV?
Sure,
of course,
absolutely.
of course.
How about you, Ben? What advice would you give?
I would echo what everybody has already said, which is just getting in touch with people that for me, it was people that I kind of admired and being like, how can I be you in the future? And the other thing that was big, and Bobak already kind of said something about this, was contacting the programs that you think you might be interested in. I wasn't a bad student, but I was no superstar of a student at Michigan. I took challenging philosophy courses and sometimes didn't do so well, and that was okay. Didn't bother me at the time, but when it came time to applying to medical school against these 3.99 GPA people, I was very self-conscious. And I remember speaking with Michelle Grundy, who works as one of our associate directors at our program, and she looked at my numbers really quick, and she was like, oh, you're fine, don't worry about it. Once it's past this point, which seemed to be very reasonable cutoffs for me, we don't even look at it anymore. And I was like, wow, that's amazing, because I thought MD PhDs would be the hardest programs to get into. But it turns out they're not just looking for numbers, they're really looking for passion, they're looking for someone who can critically think, and it was more accessible for me, and then I get the confidence to just apply all MSTP, whereas before I didn't think I could go do any. So that was a big part of my decision.
Cool. So I was sent a couple of little statements that you guys wrote to say why you wanted to be on TWiV. I don't know. I guess that was to find. I didn't select you because I don't know the students, and Jonathan and I guess Terry might have. So I just wanted to read a couple of these, not entirely, but just some of the thoughts. Bobak wrote, I'm interested in the panel because part of me has wanted to get involved in science communication. I always felt that's a major gap in the science community, discussing your work and science interest to a general audience. So do you think you'll do that in your career?
I was actually, it was interesting to hear you talk earlier today, and toward the end of your talk you had mentioned that one of the reasons you're have been increasingly getting involved in podcasts and the blogs is because you felt like you could have had a larger impact doing this than in the science. And that's certainly something that's always been in the back of my mind, and I also really enjoy writing and communicating science if I can. But it was very interesting to hear you say that, because I think A, it's something I've wanted to do, and B, I really do think that there is a gap, as you've touched on, really made clear today. And it's certainly something I want to do. I'm still struggling with how or when, and I mean, you had mentioned you just got to find time and do it, and you've also pointed out that being regular about it is very critical for maintaining a following. But quite honestly, I was going to ask you about it after the podcast if you have any tips for that. But how to kind of get involved in that, and I was envisioning it maybe later on, because I think still being a student, maybe don't have the credibility you have as being on faculty. But it's certainly something I would really be interested in.
I think you have to find something that you're comfortable with, because you're going to be busy in the next years, right?
Right.
You don't want to do something that you will then stop because you don't have the time. But I do think you have something to say at every level. So you're starting a career, you're starting in a physician scientist program, and there are people who would love to hear about your experiences on a monthly basis, if anything. Just write something, write a blog, and tell people what happens. That would be really useful.
Okay.
But I think you ought to think about it and come up with some low activity thing that you won't give up, right? Not
Right.
like podcasting, which is going to take you a lot of time.
Right.
But I do think that everybody at every level has something to contribute. The public doesn't just want to hear from old guys like me, but everyone throughout the chain, from bottom to top, through different stages of education. It's all useful.
Okay.
I mean, not everyone's going to do everything, right? So if everyone covers a little different aspect, I think that'll be fine. And that's what I would do anyway. If I were starting over, as a grad student, I'd talk about what went on this week in the lab and what happened and what didn't happen, and make it a little bit of a personal blog. That would be really interesting. I've never seen anything like that in a consistent way, and I think the public would love that. So Ben wrote that you have your own radio program on Vanderbilt's student-run radio station, WRVU, and you interview Vanderbilt faculty about their work. Is that right?
Yeah.
How often do you do that?
So this is a weekly
Nice.
program. I don't interview faculty every week because I've had more difficulty scheduling than I thought. But WRVU it's a student-run undergraduate radio station that is located on the undergraduate campus, which is conveniently about a 5-minute walk from where I work. The radio station is, so I can get there pretty quickly, and so can a lot of professors that are working at the medical center. And so last semester I got trained at the station. I got to start playing music, because the main purpose of the station is to play alternative music for Vanderbilt, which I actually enjoy doing.
So your interviews with faculty are the commercials.
Yeah. No, I actually, so the way that the show is played at this point, because I've been doing it for about a year now, or a little less than a year, and they have certain CDs that they want you to play on the radio, not necessarily sponsored, but we get early CDs from people, and it's nice to play them on the radio and tell them that we play their stuff. So for 20 minutes, we have to play, 20 minutes out of an hour, we have to play some music. The next 20 minutes will be an interview with the faculty. So recently I interviewed Larry Churchill, who is one of the medical ethics professors here at Vanderbilt. So we'll talk for 20 minutes about medical ethics, and we actually talked about communication, and then the last 20 minutes, I will ask people to pick music, if they're a faculty member, to pick music for me to play for the last 20 minutes. So Dr. Churchill was really into I believe he was a Russian folk singer who he was super into at the time. And I just played a bunch of Russian folk music for the remaining 20 minutes.
You actually had it in your library.
Well, so the great thing about the station is you can play things off your computer. So in addition to playing things off of vinyl and CDs, you can play things off your computer. So you can go on YouTube and pull up songs, or go on something like Spotify and pull up songs. And yeah, so it's a lot of fun. I even sometimes, because I've had some trouble getting faculty, I've also booked bands. So I did an interview with this band, Yonder Mountain String Band, or this big folk bluegrass band that came through Nashville, and I got to do an in-studio with them. So, but the original goal of the whole thing was to chat with faculty members for 20 minutes and just 20 minutes, I don't know what you're doing. I'm a graduate student, I'm no expert, but I can at least hold a conversation, and let's tell Vanderbilt what we're working on.
And this is live, right?
So it's live, but it is live. Actually, it's live every time, but I've recorded shows in advance and then replayed them. My show is Sunday at 6, which isn't always the best time for people. So I'll book a spot right now, at least. I'll book a spot at like Tuesdays at 3 in the afternoon. That happens to be an open spot. And then we'll do it live then, and then I'll record it and replay it for my show, which is Sunday.
So you don't put these on the web, right? These shows, they're not archived anywhere?
A couple of them are. I have recordings of all of them. I haven't put them all on.
You should
the
release
web.
them as a podcast.
But,
you could release one a week and
Yeah,
put it
I
on iTunes.
I could.
If
You're
you've
right.
got, how many do you have recorded? 20 or 30?
Oh, no, not that many. Maybe at this point, maybe 10?
You should do it. You could
So.
release one a month, you know.
Yeah.
If you want help, I can help you. You can email me. I can tell you how to do it.
Yeah,
You're gonna do it, you know?
I would love that.
Is this something you'd like to continue as long as you can?
I hope so, yeah. I've had a little bit of struggle with the radio station
Why?
because it's entirely student-run.
Damn
which
students.
means so when they go on break, the radio station goes on break. And when they go on summer break, like they will go on summer break in 2 or 3 weeks, the radio station will go on summer break. So I will be here all summer, but the radio station will be locked, which is a little frustrating for me. But it gives me plenty of time to plan for the fall.
Too bad. You could
But,
you could use it. You could do a show. You could record it and so forth.
Yeah, what I need is a setup like this, and then I can go around. Because one of the I had an idea to do a show, and I didn't have the equipment, and so the reason why I started going to the station in the first place was because
Yeah.
they have such great equipment. So
So this is portable. It's not too expensive. I can tell you what it is, and where you can use. There are actually cheaper things that you could do that are portable that plug right into your computer, even not having to have all this other wire. So if you're interested, just email. Happy to help.
yeah,
I'm
that'd
happy
be
to
great.
encourage that. So Megan wrote that you're a virologist and avid TWiV listener.
I am.
Been waiting for a good opportunity to write in, but being on TWiV seems even better. TWiV has been a great way for me to keep up with the pulse of virology while being back in my clinical rotations, which is what you said. So that's cool. I didn't see these before you were picked, so they all picked based on what they saw. So.
Excellent.
And Meredith wrote, I'd love to join the group. I can bring my perspective as a woman in the MSTP and someone about to have a big transition, moving cities. Can also talk about human metapneumovirus, which we did, right?
Yep.
So there you go. Anything else before we sign out? Did I miss anything? Is there something you'd love to get out there?
For prospective applicants to the MD PhD programs, if they have any questions, of course, feel free to email us. But I'd also just say that it's an incredibly wonderful, it's a long time, but it's a wonderful program, and I'd highly encourage people to consider it and do research into them if they're thinking about it.
Is this a good place to live too?
Yes,
yeah.
absolutely. It's changed dramatically in 5 years, 6 years that I've been here. It's just new neighborhoods popping up all over the place.
And
but
population is growing. Right?
Yeah.
It's one of those good places to live, right?
Yeah, the cost of living is still really low, but yet you're getting all these fantastic things about being in a city.
Yeah. Great music scene, not just country.
Yes.
Growing food scene.
huge food scene now, yeah.
All right, well, that'll do it for TWiV 333. This episode and all the others you'll always find on twiv.tv and also on iTunes or using any of your favorite pod-catching apps. And if you have any questions or comments and you want to ask these guys some questions, you can send them to twiv@twiv.tv. I want to thank all of you for participating today. Bobak Parang, thanks so much.
Thank you for having me.
Appreciate it. Megan Freeman, thank you.
Happy to be here.
Meredith Rogers, thank
Thanks.
you. And Ben Fensterheim. I'm sorry I messed it up. Thank you, Ben.
That's good. Thanks for having me.
I appreciate that you all took the time to do this, and I wish all of you good luck in
Thank
your
you.
time here and in the future.
Thank
and
you.
stay in touch. I'm Vincent Racaniello. You can find me at virology.ws. Any of you guys have Twitters that you like to publicize? No, you don't. You're not on Twitter, right? No blogs or anything, right?
Gotta work on
Gotta
that.
get on that. Yeah.
Okay, gotta work on it. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.